toolbox. I show you how to upgrade. So say 0. We will play with the toolbox upgrade SCGEA2 to its latest version. Then fade. This is dimensionality reduction technique. Then we will have our clustering algorithms, k-means, what are dB scan, spectrum cluster. So they all functions have been made in the statistic toolbox. We just need to learn how to code them. Then we'll spend some time with SC3. So there's a published method, and we re-implement in MATLAB. And you will look at the source code and see how the function is constructed, and follow the step by reading the paper side by side. So when you have a paper, describe the methodology, how you implement it step by step. You will see exactly how many matrices, what the transformation each step have. We open it to autonomy for this algorithms. Number 4, I will play this video, which was given by Dr. Michael Salinas, and he gave the permit kindly to us to play this to showcase how the clustering algorithms have been applied in the real data analysis. He's going to discuss how he annotated cell type in a colonocyte situation. It's very typical workflow. It's not only using the algorithm by itself. You need to have a good understanding of your system and what cell type you expect to see, and what the subcell type you expect. So this is a very good illustration, and thank Dr. Salinas. At the end, you will realize there are function in the toolbox you can call R function. So there's some built-in bridge allow you to call R directly from the toolbox. So I will show you how to set it up. Then you can utilize some of the function in R. We don't always spend the time to re-implement those algorithms because they already have a very mature R single-cell data analysis environment. So some of the function we will want to borrow directly. Okay, I will show you how to cell annotation and 5 calling R. And the next week we will show you how to call Python, okay, from MATLAB. So you don't have to leave this toolbox at all, and you're just utilizing the external function to process the data and get back the results. Still have the visualization, everything intact. Any questions? So let's open your environment here. So you know if you type this, you have the tools in there. So if you do a which, and you know this is installed in your local folder, which your own data. This folder, there's add-ons and toolbox inside their toolbox. So single-cell gene expression analysis tool is one of the toolbox you installed as add-on. So now we are going to uninstall this, okay? Then reinstall it Then in that way, you have the latest version, which online. If you don't do that, you directly install a new version. I believe this you will have another single-cell gene expression toolbox number 2 after that. You don't want to have 2 different versions coexisting on the same system. So that's why I encourage you to uninstall the older version. So how you can do it, let's click this add-on icon here, this menu. Then you click Manage Add-ons. And from your left side panel, you should be able to see recommended for you, and then you have installed add-ons. I have my lab toolbox, bioinformatics toolbox, etc., etc. And then you can find the single-cell gene expression toolbox. And then you right-click here, you click these 3 dots, and then see uninstall. So you will remove this older version, 25.9.8. You see yes. So now if you successfully uninstalled, and if you search which these 2, you should not have that in your system because I already uninstalled it. If you try to run it, you should have no function. Okay? Any questions for this step? Uninstall the older version. Yeah, so, do you have that in there? Okay, you do have, but why not showing uninstall? Okay, so this is, yeah, so this is installed, so this is the older version. You just click this uninstall. Yeah, so that one is the new version is coming in. You don't want to co-install both of them. Okay, everybody uninstalled it, and we have a clean. So to reinstall, just like you normally will do, just found this, search, so the latest version is 26.1.5. And add. Now after that, if I do a switch, and it should have this come back in the same folder. So now if you should be able to run it. Mm-hmm.
Do you have to do this for every add-on? Because sometimes you see like update add-on on the manage. So anytime there's an update, is it better to just remove it and reinstall,
Mm-hmm.
or it's just for it's package specific?
It's a package specific, so after a period of time, you should consider uninstall the old one and put a new one. There are a lot of bug fix and function enhancement, yeah, over there. Yeah. Okay, we are done with this. So now let's load example data. So we are going to play with this example data in order to run Fate. Okay, so we will try Disney, UMAP, and Fate again, and using the command line. So now we can just try to get some data back into the workspace. So you can click raw data. Okay, then click this button to export the data, the variable into the workspace, because we want to use workspace to work on the expression matrix directly. So you need X and G. So just try to click both of this and click OK. Now you go back and click this icon here. Then you should come back to the workspace, and you will see you have X here, GN list, GN name list, and also the data, which is a single cell object. Okay, so at this moment we don't need this interface because I want to show you the command line to run Fate. Okay, so we don't need this. We clean the workspace. We do a transformation to do so the variable X was in some kind of compressed version, so for the sparse matrix. But for illustration purpose, I just reconstruct the whole matrix. It will take more space in the memory, but once you click it, you can see the values. Otherwise, it will be some kind of compressed version. It's more efficient in some computation, but not good for the education purpose. That's why I converted it back to a full matrix. But for the code, for the use of the command line, it doesn't matter if you do this conversion or not. It's just data type change. So if you do this, you can see the expression matrix we get. This is real data, and that's a real GN name. So that's the two variables I want you to have. So we can play. So last week we had TSNE function. If you remember, if you gave TSNE and then you gave x, you should be able to get the embedding the coordinates. However, TSNE takes x as a rows are the samples and the columns are variables. But our x is by gene by cell, so we want to use columns as a sample. So if you really want to use this directly, you should do the transpose. Okay, you should also normalize it. So that's why I create a function called TSNE instead of original TSNE function. That just make this do this normalization itself automatically. So I will let you to try this function instead of original function provided by the toolbox, not toolbox, by the statistic toolbox, MathWorks, MATLAB itself. But this one is customized a function for single cell data to take X expression matrix, which is gene by column. So rows are the genes, columns are cells. So you don't have to worry about normalization or transformation. It will be done automatically for you. By default, you will have a 3D output. So if you look at this function, the output is S, is your coordinates of embedding of your cells. There are 3 columns, okay? This X, Y, Z coordinates. Each row is a cell. So how many cells we had? In X, we had 2,075 cells. That's how many columns in there. So the output of this function is every cell, every individual cell out of this 2,075 had been given a coordinates with the 3 numbers. That's their position in the 3D space where they are. Okay. So this S is a variable I called the t-SNE embedding for those cells. So now you can use scatter plot, scatter 3, to visualize S. This is the first position, and the second, and the third. S. Remember, those are the extraction of those matrix S and 3 columns. You put them here and give your figure. So this is the visualization of this cell has been embedded using t-SNE. Okay. The 3D. If you decide to use the 2D, you can give the extra parameter here. There's with a comma and a 2. That's the number of a dimension, and you should be able to get a 2D embedding. Any questions for this part? So in the interface, you don't have to do this manually. You should be able to just click to get this picture. But I just want to show you, if you don't click and to use this command line and manually how you achieve this. Not too hard. Just embedding, get the coordinates, and plot them. Okay. So now we change another function. Instead of to use t-SNE, we use UMAP. So the only thing I change is the function name. All these functions have been customized to take this matrix, expression matrix, and then give the output. Same kind of a framework, same calling. You just get using different algorithms to do this embedding. Okay, again, normalization and the log transformation will be done automatically. Now, then you can plot your UMAP figure here, okay? Any questions? It's okay. Again, the same story. We are going to use fate. Exactly the same, okay? Same way of calling this function and this get. There's some output on the screen, but you just have to wait. After 11 seconds, you get the result. Okay, so this is the function I called. Same way you plot them out, and that's your fate embedding. Okay. Any questions for those? So you have 1, 2, 3 different ways it's embedding, and let's see if I can tie them all so you can put them all together and side by side. So if I don't tell you which one's from which, but you somehow you can figure it out later if you use these 3 different methods on the same data set more and more. And the t-SNE gave you some kind of this cluster cloud-like. And the UMAP tend to have very condensed core like this, and they are disassociated. There's no intermediate. Fate tend to give you elongated this arms and very weird this structure. Yeah, so it's based on the algorithm behind each of these implementation algorithm. So it gives you different view, but same information from the exactly same input. And I also showed you once if you use the toolbox through the interface, you should be able to have this function you highlight. Remember last time, if you highlight this group of cells, the other group is corresponding to the same cell should be highlighted. So that's why, although you know how to run this through command line, sometimes you really want to utilize the toolbox interface to help you to have this multi-dimensional view of the embeddings. Okay. But you don't have this function directly here because these three figures are not linked. You cannot highlight this part. You want to say, okay, which the other part should be the same cell. So I don't know. Maybe it's this. Maybe it's this part. But in the toolbox, you should have that option. Okay. So now let's switch this back to Kisney. So I just want to have updated embedding S. We will do the k-means on S. So the k-means will give you is a function which we can call directly here. You can give S in there. Let's see, S. This function requires two inputs. Your input, so we are not going to use the expression matrix as the input. It requires this S, okay? S is your compressed, your dimension reduced, the 3D figure. And then see, I want to have 5 clusters, and then the output will be so I still don't know whether the k-means require the samples to be in the row or in the column. I will just very quickly to check the output. The C will be the labels, so for each cell. If this C is just 3 and give me, then that means I should transpose this because k-means may take columns as a sample. But I just want to give it a try and see whether it guys. So C output is 2005. It's all right. So C will this k-means function will take a matrix as input. The rows are the samples, are our cells, and the columns are the variables to define these rows. So so C is the category label, category tag for each cell. So you see the first cell in the C is 3. That's because k-means assign the first cell into group 3. You can browse this down and to look at the membership assignment. It should be the integers between 1 and 5 because you ask to group them into 5 clusters. Do I see number 2? So the number 4 cell belongs to category 2. If we go, number 20 is 4. Number, I haven't seen number 1 yet. Okay. Okay, to plot this, remember we we have this T-SNE embedding, but this does not give you the colors. So we need to find a way to put the color into here. If you have a number 3, I should give you orange. Number 2, I should give you a blue, and use different colors. So what we have to do is to use group scatter. I don't know if you have group scatter 3. Let's see. I don't know if we have group scatter 3. I don't believe so. This only have 2 groups. So we need to find a way to color those cells, depends on the categories. Let me think about how to do this. Let's see, scatter, this function should have a size. So this size could be, so size, so this we can put 20. So how big this marker is, and the C will be the categories, C. So if I do this to give the information C, so each cell will be giving a tag. So this belongs to group 1, 2, and this function will color it differently, depends on the membership of the cell. Is that okay, everybody? So now you can play with this K. I just arbitrarily put a 5 here, but you can have some guess how many clusters in there and replace this maybe into this 1, 2, 3, 4, 5, 6, 7, 8, 9, maybe 9, and just see whether you have a better results that fit your visual clue here. So replace this into 9 and redo this and see whether we have a good cluster results. Maybe even more. What's the best number? So there's no best solution, and I don't believe we have the optimized number here. Depends on how you understand your data. Okay, you may want to still group this tail cluster as it is, or you may want further break it down into 1, 2, 3 subgroups. It all depends on how are you going to utilize this result, okay, or your understanding of this. If you believe this is a very important, highly heterogeneous population of cell, you want to further cluster this into 1, 2, 3, 4. You can do that, okay? There's nobody can stop you doing that. Yeah.
On what basis can we determine whether that cluster is important? Like based on labeling, right?
Based on labeling later, maybe based on later labeling. So the strategy we are going to use for the annotation is to break down the cell into a lot of small clusters that annotate those small clusters individually. If they belong to the same group, same cell type, they just merge them later. Yeah. So
yeah. Further clustering will be done after cell annotation, or
no, before.
Before itself.
Yeah. So cell annotation will annotate each subgroups you clustered here.
Subgroups of the same cell type.
No, you can have a different cell type. The cluster 1 is T cell, class B, class 2 T cell, then you separate the two. If they have different names, then you keep the, not merge them, you keep the original clusters. Okay, so this k-means directly apply to embedding the results, directly apply to those points you can see. So the results are supposed to be very good and consistent with this spatial separation. You can also, of course, directly see. I want to directly class to the k-means in the original, this huge data set, into 5. Okay, this still work, and you can still have embedding. Okay, but this embedding so not as good as you expected, because you see all the clusters I see, I want to have 5 that belong to this blue group, because my k-means was applied into the original data. There's some distance computation which is not so accurate. Remember, the k-means is based on the distance between the data points. They are not a good separation. So all the blue cells have been grouped together closely. So that's why if you apply directly to this embedded, because it already help you to move the cell to the 3D space and based on their similarity, and then you get better results of the clustering. Clustering directly applied to this embedded coordinates rather than the original data. So any questions for this part? Now for the same thing, I encourage you to try dbScan. So dbScan is also a function that in the built-in function. So if you do dbScan, okay, so this function is located in the statistics toolbox dbScan function. So let's try dbScan, read its documentation, because this is slightly difficult to set up compared to k-means. K-means you just have an input matrix and a number of k. But in this case, we need 2 inputs as a hyperparameter, right? So it will also take this row-based input matrix. So we will replace this big X with this S, our embedding. And it also gave you the index, the same output as a k-means. But you need to decide these 2 values. So I will come back to here and just give it a try. See, dbScan. If I don't give anything, you say, okay, it's not optional. You need to give value of epsilon here. It has to be a number, okay? So I come back and give it to 0.2 maybe. I have no idea and how to set it up. And again, you need an integer, so minimum number of points in that neighborhood or readers. So that's 2, 4, 5, 4. I gave 2 numbers, okay? And then we make it run, and then you get those results. I don't know why they're all -1. There's something wrong with this. But anyways, I plot them out and see what's color. Looks like they're all -1s. There's one here. There is one which in the different category is 1. So this algorithm take the same input as the k-means, but it gave me a result with only a few cells belongs to the different category, and maybe put everything else into a outlier group. I don't know. So I come back to here and we'll be doing this slightly different, maybe to 0.5, and here I change it to 5. Let's give it a try, and then similar. Don't know. So it's difficult to control. I have no idea how to set up this parameter. Maybe 2 and 5. Slightly better. Yeah. So this parameter gave you completely different results every time. So I'm not going to use the DB scan. Questions? You have questions?
How can we determine what is the epsilon?
Yeah, I have no idea. I don't know. So this is a limitation for DB scan method. And then you have no control how many groups it gives. Looks like there are at least 13 different groups there. I see 12 here, and also -1. So you have somehow 13 different groups by changing the parameters here. Yeah, I don't know how they decide. So this example perform a DB scan input, and they construct, they synthesize a data set which looks like this. Okay. And then they do the DB scan with the parameter 1 and 5. Then DB scan gave you 2 clusters, and this is one and that's another one. Okay, in this application, they know this parameter works okay with the simulated data, so they just gave this as a result. You also have a selections of how can you compute the distance. And if you don't do anything here, you will have Euclidean distance. Now you use a squared Euclidean distance metric. You get the results. Same. But if you have a different, this is to show you if you run k-means on this, and you get the k-means results with this and that. Okay, so in the toolbox, through the interface, we don't have any options for using DB scan, but you can put this in your backup and see, okay, in some special case, maybe DB scan is useful. And let's see. Another way is to look at the DB scan. There are documentation. You can try to browse down to, this is one application. So each of this function, then at very end, you should have something like C also. So if you see the C also, this give you a link or pointer to some other functions which are related. So k-means or clusters data is a function. This also have a not k-means, but a k-mean noise, and linkage is for the build the trees, and the p distance, pairwise distance with the two matrices. So they're very useful piece of information under each of the function documentation. For example, if you have a k-means, if you brought it down, and you will see also the cluster and also incremental k-means. Oh, this is a new one. Let me see. Incremental k-means. It's very new, and this function was introduced last year. So you can sense this. I remember I read this. They also may have a paper, so original paper, describe this function, this algorithm. You should have the information from the reference. Okay. I don't know how this incremental k-means works. Okay, back to the DB scan. There are also documentations, the introduction to the DB scan, and it's very good review paper, basically. Introduce the history, how this, their code examples. Okay, and also because this is a commercial package, so every single details have been reviewed, refined internally. I really trust their documentation. Compared to the free packages, everybody have their own ideas, maybe not so consistent sometimes. Okay. All right, let's move to spectrum cluster. So this is a function name, and if you search which, this will point you to the statistic toolbox, which under the same folder of the k-means and the DB scan. So this is another option. So let's look at its documentation first. So exactly the same input, and X matrix, which you want to do the cluster, and the number of k. You can have distance options. If you remember, I explained the spectrum clustering algorithm. It goes to the network and the eigen decomposition and found the second smallest eigenvectors, and eventually it will be using the k-means on that one. So you go through this build of network, and then decomposition, found the feature of this network. Then you still use k-means to solve the last step. So that's why the k is required. K-means very quick, and but this spectrum clustering will help you to understand the structure of data. So apply to the case where you have this non-convex shape and still work, and because you're pre-precise the information already using the graph theory. And I also found the original idea of this secondary smallest eigenvector applications in this case is root back to 1970s. So it's not very old, okay? Not 100 years, it's something like 50 years old, the idea. Okay, so let's try spectrum clustering application to our C. So as I said, it's same as k-means. You just replace this with this cluster, and you get 5 output. And then plot them. So that's the result for this. So to us, the advantage of the spectrum clustering not so obvious in our case, because we just want to directly have a k-means to group this, obvious their clusters by I. So we know there's some kind of cluster here, but it looks like this spectrum cluster is not performed as good as a direct k-means in our case here. So what our case? Our case is the result of the t-SNE embedding. T-SNE embedding by itself already try to group those cells into very obvious clusters. There's no very weird shape like those non-convex shape. So, so this is not the best user case to spectrum clustering. You see the result is not as good as we hope it. I will give it a try to the original matrix X transpose. This is supposed to take longer because you have to compute the network distance and no good. So eventually, so we compared this. So eventually in the interface, the toolbox, we choose to use this directly applied to t-SNE embedded cells. That's what we are going to do. Not options. So any questions? No? Okay, clear. Yeah.
There are two of them. What's the difference? One says spectral clustering.
Yeah.
One says spectral cluster. Are they the same? When you
Oh, the function.
yeah, the function.
Another one. Good question. So you type this. This is one option. Spectrum. You see this one? Yes. Yeah. So if you, I believe if you do a which, and this is a function, yes, I would like to know which, where this function is come from. It may be, but I know this is from the statistic toolbox itself. If you do which and this is a, maybe it's object, something in there. Both from this, okay. Yeah, so this will be the help data structure for. Good question. So this is add-ons toolbox. That's okay. This is my toolbox. So this one is a community-based. Yeah. So it's not official. So if you look at where this function is from, It's in the add-ons in my toolbox. And inside my toolbox, there's external. So I just borrow someone else in this implementation. So if you search Google to this original software, you should be able to find who developed that algorithm. Yeah. So if you don't install the toolbox, our toolbox, you don't have that option. Yeah. Okay, so now let's take a 5 minutes break. When we come back, I will show you play this video first, then we move. Okay.
So it's really the identification of all the different cell types in the mouse colonic tissue. So there are some data processing steps. The first one is filtering based on the QC plot, and I'll talk a little bit more about that and how I decide what to filter. Then it comes to the identification or the separation of colonocytes versus the non-colonocytes. These non-colonocytes are really just going to be the immune and the stromal cells that are also in these samples. And then once I can separate those, I can focus on the colonocytes, and I can then start to try to identify all the different subtypes of colonocytes in these crypts. And I'll talk again more about this in a bit. So this is the colon tissue. So we isolate this colon tissue. This is a human, but we get it from mice. And we have now started using the whole intact tissue. And what I mean by that is before we were just kind of taking off this top part, this epithelial layer, and then sequencing that. But we do know that down here is in the muscle layer is where all the blood flow is, all the immune cells are, and they are also responding to things like diet. So we're now interested in looking at the whole colon tissue. So we now have all tissue there. This is kind of what it looks like at a cross section. At the top here, you have the epithelial cells. These are the colonic crypts that are right here. And then down here, this is where you have your immune cells, your blood vessel cells, muscles, all different kinds of fibroblasts that give nutrients to the colonic crypt stem cells at the bottom. It's all there. So everything is important, and I think it's good that we have started doing this, but it does make the data a little bit more challenging. And as you can see within these crypts, there are a ton of cells really squeezed in here. And even in the center of the crypt, some of these circular things are part of goblet cells. So if we use a different staining, we can kind of see that the blue here are those goblet cells. And so we can capture all of this with our single cell data. So we have all of these different cell types here in the colon. This is just a simplified version of that. And this is what we've been using to do some RNA-seq. So the first thing, like I mentioned, is the QC filtering. This is really going to depend on whether you're using Flex or the 3' sequencing. The Flex is really clean data. You almost don't need to filter this data. If you do filter it, it would be just to kind of get rid of these cells that have a high mitochondrial percentage. But everything else here, to me, kind of looks fine. But if we come over here and we look at the 3' expression data, if we just look at the number of genes, this graph here, just from my experience, I've learned that down here in this section is really kind of junk cells. So we want to kind of filter this out to where it looks a little bit more like this over here. Even in the number of reads here, where there's this break right here, below that is kind of just these junk cells that, again, that we need to filter out before we begin processing the data. And that's how I filter my data. I have to look at the QC plots and kind of just go from there. Again, the Flex data that I'll be showing today is pretty clean, so I actually didn't perform any filtering on this data. This data comes from notobiotic mice, that is mice that don't have a microbiome. So this is pretty much just pure colon tissue, unaffected by the microbiome. This is what the data looks like when we load it into Dr. Sai's software, and then we can begin to embed it. I like using the UMAP because it just makes a lot more sense to me biologically compared to the other embedding methods that you can use. So the first thing when we have this plot, like I mentioned, is going to be trying to separate those colonocytes from the non-colonocytes. I use two major markers for this. I use EpCAM and EcadHerin. These are going to be expressed highly in epithelial cells. And this is what I use for me to separate the colonocytes, which are EpCAM positive, from the non-colonocyte cells over here. Of course, this isn't enough. I have to go in and really dig deep into what these cell types are. So just so that you guys know, about 60%, it's really about 50 to 66% are colonocytes, and the other percentage, 34% is going to be these non-colonocytes. And again, this is going to vary. These are mice that don't have a microbiome. So in the normal wild type mice that we do see, this is going to change a little bit. There might be more immune cells because of the microbiome. I'll just really briefly talk about the non-colonocytes, but it won't be my main focus because these cells are actually pretty easy to identify. You can see that they make up just these little clusters here and they separate really nicely, and we can identify them And we get all different sorts of immune cells, B cells, macrophages. Sometimes I see dendritic cells, sometimes not. There's also blood vessel cells, so endothelial cells, smooth muscle cells, mural cells, and pericytes that are all connected to the blood vessels. Then there's all sorts of fibroblasts, some myofibroblasts, and then some other types of fibroblasts that, like I mentioned, are providing growth factors and nutrients to the colonic stem cells. And then we also see things like neurons and even adipocytes show up in this data. This is just a heat map for all of these cell types, so they do kind of break out really nicely. And I'm pretty confident in these cell types too, but I haven't put a lot of focus into this. I haven't really analyzed data coming from the non-colonocytes just yet, because my major focus has been on the colonocytes. So this is when I subsetted out the colonocytes, and then I begin to try to cluster these cells and see what are these cell types. So I use k-means clustering, and I kind of just play around with the numbers until I find one that really separates out everything kind of the way I like it, like visually, and then I can kind of go from there. So the first thing I do with the colonic cell types is going to be separating out the enteroendocrine cells and the tough cells, and that's just because they actually stick out from the data. So in this case here, this cluster 11, I know is going to be the enteroendocrine cells, and this cluster over here, number 3, is going to be the tough cells. And I know that by the marker genes that they're expressing as well, and we can actually calculate a score for these marker genes for the enteroendocrine cells. So you can see here they scored really highly compared to the rest of the clusters. That's cluster 11 up here, so I know those are enteroendocrine cells. And the tough cells, again, scored really highly and different from the rest of the cells. That's number 3 right here. So these guys are tough cells. That's easy. That's actually pretty easy and shouldn't be too much.
It's hard to me, but
like again, it's just because they actually pull away from the rest of the cells, because they are a differentiated cell type, but they are technically a specialized cell type as well.
Expression is so different; they just separate from the rest.
Exactly. The other cell types that I can try to pull out are goblet cells. Goblet cells are going to be this whole chunk right here. And I know that based on the MUC2 expression, so you can see that it expressed very highly in these clusters here. So I know those are goblet cells. I can also calculate a score for them, and they do separate out compared to the rest of the clusters down here. So I know, again, these are goblet cells right here. So I can actually subset out the goblet cells again, and then begin to focus on the specific goblet cell subtypes. I know nobody really does this, but I thought it was very interesting because once I saw this, I remembered a paper that I had read that actually mentioned that there's 2 lineages from the goblet cells. There's a canonical and a non-canonical lineage, and that's actually what I observed here, so that was pretty cool. Down here, what I'm showing is MKI67. This is a proliferation marker, so you can see that the clusters about right here are going to be the cells that are proliferating.
so that's interesting because they're supposed to be generally quiescent, right?
Yes and no.
So there are actually some cycling cells in there.
Yeah.
They're going to divide. Interesting.
There's also differentiation markers. So for goblet cells, a good differentiation marker is MXD1. And you can kind of see that, well, yeah, I didn't do a good job of lining these up right, but down here are going to be where those proliferating goblet cells are. And actually this little cluster right here that you see is secretory progenitor cells, or what I think are secretory progenitor cells. And then we have the non-canonical goblet cells here using these 2 markers that I grabbed from that paper. So this kind of branch right here is non-canonical goblet cells, and these over here are canonical. You can kind of see that this non-canonical branch is actually very close. That's this branch right here. It's very close to the other colonocytes right here. So it was very interesting to actually see that out on this 3D map. But based on those markers,
That's a biology.
yeah.
That's right.
To us, just 2 branches. We don't know.
Yeah, I know, and I
We needed to read it.
I remember that paper. So for the secretory progenitor cells, they have different names and people call them different things. That's what makes this also very complicated. So I started calling them secretory progenitor cells. I believe previously we were calling them deep crypt secretory cells. So they do have this Reg4 marker. You can see that cluster number 12 is scoring really high for this register marker. That's this cluster right here. And you can also see that clusters 10 and 12 are also proliferating. This is a proliferation marker, so I know these are proliferating right here. DLL1 and DLL4 are expressed again in these secretory progenitors, and you can see that cluster number 12 is scoring high. It's actually the only one that's kind of scoring. So to me, these are secretory progenitor cells. This is what it looks like once you actually apply those cell types. So we have the secretory progenitor cells here. We have some proliferating goblet cells here, and then the rest, even though there's two lineages, I merged them just to simplify things. Those are the differentiated goblet cells. And actually, if we look at the differentiation potency, you can see that the secretory progenitor cells score really high in the proliferating goblet cells, and then the differentiated goblet cells at the bottom. So this looks good. That's what I expect from this data. Like I mentioned, we have these differentiated cells, but they're also very specialized cells. They have nice functions. And so you just saw that I was able to identify all these cell types here. The next thing would be to identify the stem cells and the absorptive colonocytes. So you can kind of see from this image to this image, I got rid of all those specialized cells. And now we're really left with the backbone of what I guess colonocytes do. So the colon is largely responsible for absorbing any sort of water that the small intestine didn't absorb. So that's what these cells, that's their function, and that's why they're called absorptive colonocytes. And so what I did was I subsetted out the stem cells and the absorptive colonocytes. They form kind of like this gradient, very similar, I guess, to this crypt right here. They form this gradient from stem cells and transient amplifying cells down here all the way up to differentiated cells over here. So I know that because of LGR5 expression is located down here at these clusters at the bottom. You can see that these are also the same cells that are proliferating based on proliferation markers. And then there's some differentiation markers that I use. I like to use KRT20 to identify the absorptive colonocytes. And aquaporin 8, this is, of course, a receptor that is responsible for pulling in water into the cells. And that is what I use to identify the late absorptive colonocytes. I don't believe I actually have an image of that, but this is what I'm currently working on right now. This is what I've had to do to be able to identify these cell clusters. My initial thinking when I first started all this single cell stuff was that, we'd be able to have our cell atlas with all our gene markers and cell types and just kind of, click and play and it'll identify stuff for us. I learned over a year or two that is not possible at the time, especially not for this particular cell type. It works actually pretty well for things that separate out really nicely, like blood cells, like peripheral mononuclear blood cells. Those do separate really nicely into T cells, B cells, NK cells, and I can actually identify them using a cell atlas. But for the colon, I just haven't been able to do that. So this is what I have to do. I just have to look at the expression across these figures here and then try to label these clusters here as a specific cell type. Again, this isn't finished just yet. I need to input a lot more details into this so that somebody can actually follow it. But for the most part, this is how we identify cell types in the colon.
So that's why, so you have potentially another approach that you were trying to optimize.
Yeah, I don't know the exact step.
We don't go, this is assimilated this, which makes it worse. Are just, like you said, the immune system it's the truth for normal colon. And the other thing is very novel, meaning that no one has ever created an atlas for a notobiotic mouse. And here he is, Michael's done it. And then nothing changes from the atlas standpoint. And then you can move into any other complementary assay.
Yeah, so this is how this cell type annotation manual, cell annotation should work. You have to be very familiar with the system, so you know what cell type you expect to see. And you'll be able to have this picture on your mind, the marker genes for each cell type. So you know, okay, I would like to look at epicam, and you know, okay, that's separate epithelium cell from non-epithelium cell. So that's epicam, the single gene can help you separate them. And then you have to go inside. You know, okay, this is a differentiation that's a node, and which stem cell the index. So you have to have those knowledge. The overall is we cannot rely on this so-called cell atlas. At first, in the community, people want to build atlas, so we have all the information about all model system, all the reference cell type there. So we just need to compare our cells with this reference. If it look like epithelium cell, then give the name. If it look like goblet cells, but those atlas you build is from other experiments as a reference. Sometimes it works, but then sometimes not. So you need to have this very detailed sub-clusters of a cell group and work on each small group one by one and look at the markers and look at, okay, those subgroups should be merged, considered as one branch, and another should be another. So Michael just demonstrated very well in this case how this cell annotation should work. So that's a very valuable exercise, and I use this as an educational tool to help you to understand how, at this stage, this manual annotation should be done. Okay. I hope it answers a lot of your questions, and I will come back to examples. Let's do one examples here. So let's load this example data which not processed yet. So let me change this color. Okay. So what we can do first is to do the embedding. I will use 3D T-SNE as an example. Okay, from here, there is a function, let me see, assign cell type to selected cells. You can use this, for instance, you click this and you start to brush. You should be able to select the cell. Let's select the cell first. So you select this group, okay, using the brush tool here. And then you say, okay, what's the type of this selected one? And you should have a very quick options. Say, okay, the top one, how about this? So this function can give you a quick understanding of what general cell type you should have in your sample. Let me see, is it human or mouse? This is human data, so I select the wrong species in my annotation. So this is a function to help you to quickly, if you're curious about what the cell type of this particular group is, you just brush it and see very quickly get the annotation. This top annotation gave this group endocrine cell, and the second option is alpha cell, and then the third option is particular annotation. So those are top ones. Then you assign this. Okay. So if you want to annotate all of them, you don't want to do this manually one by one. So what you can do is to first to do this cluster. Okay. If you look at this 3 bubble icon here, it will help you to cluster the cells using the embedding. That's what we just learned from the k-means, and it will use this as the input. If you have a different embeddings. You have a fate, a UMAP, you can select differently. But now we use this as the embedding input, and you see like k-means. Okay. And here you see like how many clusters you want to make. This number is all pre-computed based on how many cells you have. I will group every 50 cells into one cluster. So I just divide by the total number to 50, and so at the end you will get a lot of small clusters. Each contains about 50 cells. Okay, so this is a strategy to over-cluster your cells into many, many small groups. I know it's over-clustered, but I just want to do this. Then by small clusters, we are going to run cell type to each group. So after you cluster, you click this button here, and this will search a database using marker genes to assign each cluster a name. So we will select the species, and the top you assign automatically. So you know how to do it manually, select this, but now we just automatically do all the 20 clusters altogether. So this procedure will go and pick the top one and assign the cell type. Then you have to merge. Okay, if you have fibroblast 1, fibroblast 2, you can see if you don't merge and you have all these 20 different clusters, because you cluster them into 20, and they all have a different name with an underscore. This is group 19. This is group 20. So this group 8, group 20, they are close to each other. They have the same name. They definitely should be grouped together. So that's why once you finish this automatic annotation, normally you just want to merge the same name into one group. So you name them, group them, then you have a final annotation, this automatic annotation. So to give you the foundation for your next step, manual refine of this. Okay. If you don't want to do that, you can have all the individually clustered, and then you look at each subgroup, slowly merge them. Okay. Yeah.
If you have your own curated marker genes,
yeah,
can you like create like your own database?
Yeah. There's a place which you can see. Okay, I have a list of marker genes. This special cell type nobody have ever studied. There's not in the database. It's fully customized. Let me see where the function is. So there's annotation. So you see annotation. Yeah. So the second option, so you should have these options to use your own customized marker genes. So here, I'll give you an example. So I have a customized, see, cell type 1, cell type 2. Then these are marker genes. So this is just randomly generated as an example, but you can prepare your own. Okay. Then you click this, and then so the cells will be annotated based on your customized. See, okay, this is a cell type 1. The rest, and there are some unknowns. They are not assigned. So you can do that in here. Okay, any other questions? Okay, so this button is to use k-means to do cluster. So the input will be the position of the cells. This is like a shortcut, and then you can also see cluster using cell embedding. So in the menu, this item here is the same as the button you click here. So embedding the cell using the cluster cell using their embedding. However, if you want to do SC3 will, so this algorithm, remember we mentioned, is not using the embeddings. It starts from the big expression matrix directly to compute the sense matrix and do the transformation. It's a time-consuming process. If you decide to do that, this is an option which allow you to run cluster cells using expression matrix directly. Okay, that's the only option we had. I put 2 turtles here because it's slow. Yeah, in this case, I want to have 5 different clusters, for example. So that's the place where you can use SC3 as your clustering strategy. What we found is in single cell data analysis, this is not very useful, but in the spatial transcriptome, it's very, very useful. Okay, to cluster not the cells, but you have spatial information, the probe sample, the different location of the tissues. That will do a good job in this case. Okay, so that's your cluster results using SC3. Again, that's not straightforward because the input for this is the expression matrix itself. It's not considering how you pre-process your data using the Kisney and where you put in this 3D space. So this algorithm will not respect to any of this location of the cells. So your result may be not as good as you expect to see. Okay, this is a clear cluster because SC3 does not require consider this as a separated cluster. So if you want to change the colors of those cells, you can click this label here. Okay, there's a small tag in the second row of the button here. You select, say, I want to visualize the cell type. So that's your cell type. You just have this customized data you annotate. Any other questions in this case? Now, let's using SC3. Okay, so if you decide to use SC3 using command line, that's the way to write. So that will call a function in the subfolder in the run, and then ML stands for MATLAB. There's some R function, it will have a R underscore something. And if a Python version, you will have a PY underscore. But this is a MATLAB version of the SC3. We can run this, and you already tried in the interface. Through that menu, you can run this, but that's the code behind that. Remember the procedure we had, the 3 distance matrix, and then transformation, and then the different range of the chopping, and then, yeah. Go back, yes. There's a lot of output. So this is a command line, and that's a step 1, compute the distance matrix, compute the Spearman distance, Pearson, then do all this, and then after that assemble this. So the results will be a number of each label of each cell. Okay. So C is no difference from a K-means, from DB-scan, the same structure of this. And you get this indicator of the groups. Okay, now I want you to try to open this implementation, and the way to do this is edit, and then you see the source code. And you see the reference, the paper. Go below, you see this the process of inputs, and then we need a K and the number of the clusters. Normalization, transformation will be done. Here is to compute Euclidean distance matrix. So you get a distance matrix, squared form. Okay. Then you run this, get the class. Then use Spearman, another distance matrix. This Pearson, so through the correlation computation. Then there's cluster assembly steps. There are Laplacian transformation, there are PCA, there are 2 transformations, remember, in the algorithm. There are K-means, and through this sliding different range, and then combine. So everything is transparent, and you can see the implementation of the original algorithm. Okay, nowadays you probably don't have to do this. Now you can have this special prompt give to ChatGPT or Cloud. See, I give you a paper, and read this paper and implement R version or MATLAB version of this SC3 algorithm. You can give it a try. You just drag this PDF into it. It will give you a fully implemented source code, and oftentimes it works. But before that ChatGPT age, we have to re-implement everything from scratch. Okay. Okay, so we are done with this part. We still have 20 minutes to see prepare calling R. So let's let me show you the interface. If we have a data here, their external function. There are tools and Python. So there's sub-menus here, and you can run, for example, using Surat to do this UMap to transformation. Okay, their options rely on the R environment. So we need to set up our environment. So when you click this button, the program knows where to save and call the R. So those functions, for example, the pseudo-time analysis, we are going to not going to re-implement in MATLAB. So we rely on this external package. So let's see, you need to, in order to run those R functions, you have to have R installed on your machine. So if I do R Studio, it's not here yet. I don't have R installed on my machine, so I do need to go to the Software Center. Here, Software Center, to install. Yeah, we need to install R. Yeah, I hope this R Studio will come up with R together. So I just try to put R Studio in my local machine. If you have already had that, you don't have to do. You just need to know where you installed R. Then we will in the MATLAB to make a link to know, okay, your R is installed there, and that's all we need to know. Okay, looks like it's already done. So let's found. So I can click my R Studio. Yeah, I do need to install R itself. So I don't have R installed yet. Yeah, I don't have R in here. Do you have R on your machine? You do? Okay. I don't. So I don't have our installation provided on this machine, so I cannot do it. But your machine has R, that will be good, and your own computer, you have a administration privilege, then you can install. Let me see if I can work around with the store. Okay. Well, this is not a reasonable language. Really. So I don't have R installed. I will contact my IT people and see can help me out later. And R is not provided as a environment in Microsoft Store, so I cannot install it. Oh, maybe I can download from from a website directly and hope to run locally. Yeah, I'm just trying to install my very basic setup of R for myself. So I need to remember where I installed. So this is a location which I will need this folder and later on to let MATLAB knows where I installed my R. Okay, good. So my R is here. Okay, my R is here, so I'm okay now. Just turn everything off, come back to my MATLAB. So we have R, now we need to set up R to link this. So you move your options to set up R environment here. Then you will have to locate where you installed and to find this rscript.exe file. So I know where I installed, so I can find it. I know I installed in programming R 4.5.2, so I go into here in the bin and found in bin. I found this script. Okay, now I'm done. This function I suppose be able to use. Let's try easy one to use this surrogate embeddings. So I'm going to run this surrogate workflow. And you can see the source code is in R. And you can run, need a 2D. The first time you may have to, the program may install the surrogate package. Take some time. And then next time you don't have to reinstall surrogate. You should be able to have a very quick response. You see behind this window, the R is running at the end, backend trying to install for the first time the Surette package, and then now it's using this R to do this embedding. Okay, so this is a embedding generated by R Surette package. Okay, I think that's the demonstration of the use of this. So next week we will install Python, and then you have all the options for the Python tools have been already integrated into this toolbox. Okay. All right, so that's all for this week, and let me know if you have any questions. Otherwise, I will see you next week. We will discuss the clustering algorithm, classification algorithm, not clustering. Clustering is done. Okay.